Hello, I'm Timothy Weaving, a PhD student at University College London, and I'm pleased to welcome you all to the 26th instalment in CompBioMed's eSeminar series in collaboration with the Virtual Physiological Human Institute, or VPHI. The talk today will be given by Markov Viceconti, Professor of Industrial Bioengineering at the University of Bologna, Director of the Medical Technology Lab at the Rizzoli Research Hospital, and founder of the Insignio Institute at the University of Sheffield. He has a PhD in bioengineering and has been developing in silico medicine technologies for over 20 years. Before we begin, I'd like to briefly remind you that the session today is being recorded and will be available on the CompBioMed website and YouTube channel shortly. So please do use this to go back over parts of today's talk and others in the series, and maybe even share these e-seminars with your friends and colleagues. Now, there'll be a Q&A session at the end of the talk, so please save your questions until then. Once the talk is finished, you can post your questions in the Ask the Staff a Question panel, which is accessible via the question mark icon that you should be able to see on your screen. Marco, if you don't mind going on to the next slide for me, so that the audience can see what that should look like. So then you can go ahead and post your questions in there, and then I will read them out to Marco to keep the session running smoothly. Now, that's everything from me. I very much hope that you enjoyed today's talk, and without further ado, I'll hand over to you, Marco.
Thank you very much. Can you confirm that you can hear me? And
yes, I can hear you very well.
Okay.
Yes, slides visible.
Good. Perfect. Good afternoon, everyone. We're going to discuss in detail today one particular type of in silico medical application aimed to evaluate the efficacy of new pharmaceutical treatments for osteoporosis, and in particular reflect on how this type of large-scale simulation needs high performance computing in an almost desperate way, as we will see. Okay, so for those of you who are not into this particular disease, osteoporosis is the progressive and pathological increase of the porosity in the extracellular mineralized matrix that form the human skeleton. Our bones are porous, but as we age, this porosity tends to increase. And up to a point, this process is physiological, it's related to aging. But in some cases, this happens with a pathological speed, and this is a disease, and it's called osteoporosis. The major effect of osteoporosis is a silent disease. You can be severely osteoporotic and have absolutely no symptom whatsoever until you have a minor trauma. And a trauma that normally would produce absolutely no effect cause a bone fracture. The most common site of osteoporotic fracture are the wrist, the ankle, the spine, but probably the one that has major impact on the health of the patient is the hip fracture, the fracture at the proximal femur. This is an X-ray of a patient who had experienced one of such fracture, and you see how the femur is being almost decapitated. You need to intervene surgically. There's no way that this fracture will heal by itself, so you have to intervene surgically. And the effect on the elder population are usually quite severe. About 20% of the elders who experience a hip fracture will die of related complication within the following 12 months. And of those who survive, about a third will remain with some physical disability of different level and kind. There are quite a few drugs available to treat osteoporosis, but none of them solves the problem, in the sense that the absolutely best performing drugs reduce the incidence of fragility fracture of this kind of between 40 and 60%, depending on the clinical studies. So that means that the best case scenario, there are still 40% of fracture that happen even if you take the medicines. And that's the reason why only in 2019, we reported over 3 million hip fractures in the European Union only. So obviously, there is need for better treatments that reduce even more the incidence of this devastating event. So why is that currently there are no drugs in phase 1 clinical testing? So there are no new molecules being tested clinically. Why is that? Well, the reason is that the clinical trial required to bring to the market one of this drug became too expensive. You have to consider that about only 4% of women over 40 will experience a hip fracture in a period of 5 years. Okay, so already the incidence of the fracture event is quite low, but regulators are still demanding in many cases that the pivotal trial, the big phase 3 clinical trial, is done against placebo. Now, because there are drugs that mitigate the risk, the only ethical scenario for doing a clinical trial with placebo is to use to enroll patients that have very low risk of fracture. Okay, so this number, this 4%, is even lower in those cohorts. So as a result of these two factors, in order to observe a few hundred fractures, you need to enroll something in the order of 10,000, 20,000 patients and follow them up for 3 to 5 years. I mean, so we're talking about a clinical trial that could cost more than 300 million euro, and this is way beyond what many pharma company, even big one, are ready to spend for this type of products. Now, one may wonder, why don't we use computer simulation? Almost any other industrial sector use computer modeling and simulation to assess the safety and the efficacy, the functionality of complex products like airplanes, nuclear power stations. So why is that in healthcare we keep testing things by trial and error? Because a clinical trial is a trial and error study, okay? Well, maybe the question is, but can we trust model to predict with good accuracy changes the health status of patients? Well, I think we can say yes at this point. I mean, here I listed 6 examples of success stories reported in the literature in the last few years, where patient-specific models were used to predict with excellent accuracy changes in health status in various different conditions. So can we use modeling to address this unmet need related to osteoporosis? Well, if you have a model that predicts a binary event, fracture, no fracture, there are essentially 2 ways to validate this model. The first is to use a receiver operating characteristic curve, which allow you to calculate the so-called stratification accuracy, the ability of a predictor to separate correctly between fracture and non-fractured cases. And this is a very effective way to compare 2 predictors, okay? The one with the higher area under the curve has the better stratification accuracy. But to have a true prediction accuracy, you need to make prediction and then observe and see in how many cases you were right, okay? And that requires a fairly complex design to perform this type of validation. Are there already predictors of risk of fracture? Oh yes, plenty. The problem is that they are not very accurate. There are biochemical biomarkers that are correlated to the risk of hip fracture, but their stratification accuracy usually never exceeds 60%. Now, if this sounds like a good number, think that if being a binary decision, if you toss a coin, you would be 50% correct, okay? So half of the time you would be right, statistically speaking. So 60% is not that much better than the random choice. There are statistical models based on epidemiology data. One fairly popular for many years is FRAX, that incidentally was developed at the University of Sheffield in the UK, where I worked for many years. But even the best performing of these statistical predictors usually never exceed 65% of stratification accuracy. And then there is what is considered clinically the gold standard. There is a special type of X-ray machine called dual X-ray absorptiometry, which can be used to do a very low dose image of the region of interest, for example, the hip, the region of the hip, and then use this picture properly calibrated to calculate the areal bone mineral density, and a quantity that expresses essentially how much bone tissue is left in the skeleton in a certain region, okay? And there are different studies that suggest stratification accuracy ranging between 65 and 75%. And while this is considered good enough to support the clinical decision on individual patients, regulators believe that this is not good enough to use it as a predictor in place of a randomized clinical trial with fracture outcome. In the following, I will refer to two clinical cohorts that we have been using and we're using for the validation of our technology. The first is a cohort that was collected in Sheffield, and it's a retrospective cohort. It's 100 women. Half of them had a femoral neck fracture just a few days before the CT scan. And the other half are women who had no hip fracture, and they were selected to pair match the fractured women by height, weight, and age. So for each fractured woman, there is another woman who is not fractured, has the same age, weight, and height, okay? On the contrary, the Bologna cohort is another cohort of 100 women, but in this case, they were randomly selected, and they are all postmenopausal women, typically over 50, but they may or may not have fracture in the 5 years after the CT scan. And in this case, only 4 of these 100 women had a fracture in the following 5 years. Okay, so the technology I'm going to talk to you today is called BoBCAT, which stands for the Bologna Biomechanical Computed Tomography. Is the digital twin technology. What does it mean? It means that I take data of an individual patient, in this case, computed tomography data, CT scans, properly calibrated, so the so-called quantitative computed tomography. And I use a number of techniques that we develop over the year to transform this into a heterogeneous finite element model of the patient femur that can be used to predict if a certain force applied in a certain direction will fracture the bone or not. These are computed tomography. They are based on X-ray, so there is an ionizing radiation that is given to the patient, which introduces some limitation, although the most recent technology has drastically reduced the effective dose required to produce high quality images. And we generate this after the CT scan has been calibrated with a special phantom so that every pixel in the image can be translated into mineral content density, okay, and tells how much mineralized tissue there is in that voxel. After the CT scan, we have to segment the femur in order to extract the geometry. This was done first manually, now is being done routinely with the semi-automatic techniques, but we are currently developing machine learning-based techniques that should completely automate this process. The geometry is then meshed into a finite element mesh using parabolic tetrahedrons. And then there is the problem of heterogeneity, that within the femur, there are various different zones that have different porosity, which at the macroscopic scale translate into a different module of elasticity, okay? However, this porosity correlates to the apparent density that is measured by the attenuation of the X-rays. So by looking at the grayscale, of the properly calibrated grayscale of the CT images, we can estimate the module of elasticity of the bone tissue in that particular region. Of course, you have to remap the CT images onto the finite element mesh, and we developed many years ago software which is available publicly as a freeware that do this, and this is being extensively validated over the year. How did we validate initially this model? Well, comparing to experiment like this. One advantage we have compared to other domains is that the biophysical and the biomechanical properties of bones do not change significantly post-mortem, okay, as far as, of course, the bone is properly preserved. So you can use cadaver bones, you can CT scan the cadaver and then dissect the bone and then perform biomechanical testing like the one you're seeing here, and compare the prediction of the finite element model with the measurement of the strains or of the force required to fracture the femur. You're not seeing 3 femur, you're seeing 1 femur and 2 mirrors, because the other validation we can do is to compare the point of initiation of the fracture with the point that the finite element model predicts to be the one at maximum strain. If you look at the time scale, this is high-speed cinematography, you will notice that the fracture propagates almost entirely through the femur in something in the order of 2 milliseconds. So in this particular instance, we can assume this material to behave as a fragile material and neglect the post-elastic behavior. This is not true in general. There are other bones that fail in under different loading conditions where you observe a significant post-elastic component. So it's not a general truth, but it's true for this particular case. This makes possible to use a relatively simple failure criterion based on strain, which allows you to predict if a certain level of deformation induced by a given loading will cause fracture or not. 95% of the hip fracture occur because of a fall, okay? And the falls, the way elders fall, as you can see here for these 4 examples, are very, very different, okay? So it's not really possible to make a deterministic model of falls, but we can elaborate a stochastic model of falling by looking at certain constants, okay? For example, in the vast majority of falls hand on one side. Even the fall is initiated frontally or posteriorly, there is a protection instinct that tends the person to rotate the body while it's falling so that the impact happen on a side. It might have very different direction of impact, but tend to be always on that region, okay? Truly frontal or truly posterior impact are relatively rare. that means that we can idealize a body falling on a side with an inverted pendulum. And then put all the complexity that we just saw in those 4 movies, in which, you know, every elder actually felt in a completely different way, into what we call a postural attenuation coefficient. So a coefficient that reduces the worst case impact force to some extent, accounting for the particular postural strategy that the patient has adopted, whether by bending the knee and so lowering the center of mass while the patient is falling, or by discharging some of the energy against the wall, against an object, against the Christmas tree in one case, and so on, okay? So by combining all this, we come to a composite model. So there is a stochastic inverted pendulum dynamics that calculate the impact force depending on a number of statistically defined parameters like the initial angle, the initial velocity and the initial acceleration, and the final impact angle. On the other side, there is the finite element model that for a given force and a given impact direction can tell you whether the bone will fracture or not, and what is the deformation induced in the bone by that force. In the middle, there is the transformation function that say, okay, of this impact force, only a certain amount will be transferred to the femur because the rest will be absorbed by the elasticity of the floor, by the clothes that are wearing, and most important, by the soft tissues that are wrapping my hip, okay? And we found that this parameter is actually very important. So we now measure the thickness of soft tissue in the CT scan. So this is the patient-specific information that we feed to the model. By putting all together, we end up with a multi-scale model that has 8 stochastic inputs, 4 patient-specific inputs, which are the calibrated CT scan, 8, weight, and the soft tissue thickness. We derived the probability distribution for a stochastic variable from the literature, and then we sample everything with the Lightning upper cube Monte Carlo scheme, and then we should run the finite element for every possible fall simulated. This would become computationally too expensive, so we interpone between the 2 models a surrogate model, and we found that we need to run the finite element model only 20, 30 times to have a sufficiently accurate surrogate model. So the result is a system that takes some initial condition, the height and the weight, the CT scan, predict the distribution of possible fall forces, and then for each of this fall, predict whether that patient will fracture or not fracture. And in the end, we can calculate the absolute risk of fracture over fall event by calculating how many fractures cause fracture, how many falls cause fracture over 1 million of simulated falls, okay? And this is the parameter that we call RF0, absolute risk of fracture at the time 0, which means at the time when the CT scan was done. Now, is this predictor better than the clinical standard? It took us quite many years to demonstrate, but the answer now is yes. The absolutely best performing clinical parameter has an accuracy of stratification on the Sheffield cohort of 75%. BoBCAT has an accuracy of 87%, so a 12% more accurate in terms of stratification accuracy. We also did, of course, as I was mentioning, an extensive technical validation that involved also cadaver femur. This finite element model can predict the strain induced by a given force as measured by strain gauges in various regions of the femur with an average error of only 6%. They can predict the force required to fracture a particular cadaver femur in a particular direction with an accuracy of 85 to 88%. Okay, so this is the model. What is the computational cost of the digital twin, of BoBCAT digital twin? Typically it takes 100 core hours to run the whole simulation for one patient to predict the RF0 that I already defined. And we also predict something called the minimum side fall strength, which is of all possible direction of falling, which is the one that produce the minimum strength, and how big is this minimum strength, okay? There are some nonlinearity involved with it because we simulate a nonlinear context. So, and the finite element model is relatively big. It's a 3 million degrees of freedom finite element model. But I mean, overall, we're not talking about a supercomputing problem here. But what we're trying to do in Bologna is to move from the digital twin, so a tool that you can use to predict the risk of fracture in an individual patient, to an in silico trial. And the in silico trial solution, we call it BoneStrength. How can you use an in silico simulation of an osteoporosis clinical trial? Well, you can use it almost at every step of the drug development cycle, from the very early cases where you have only preclinical evidence of efficacy, and you can try to translate this evidence as how many fractures less that particular molecule will produce if it is given to the patient, up to use in designing clinical trials and then many other applications. How do we transform a digital twin into an in silico trial? Well, one possible way is this. So this is the BoBCAT. It takes weight, height, a CT scan, and the segmented geometry, and predict minimal side fall strength and absolute risk of fracture, okay? Now, this black box has a certain number of inputs, okay, that we call the feature set for patient X. And in general, digital twin is a model that, given a feature set for a patient X and given a certain condition, for example, a change in the treatment, will tell you what is the response of patient X to treatment Y, okay? Now, what is an in silico trial? Well, you look how this feature set change between patients. You build probability distribution for each of the quantities in the feature set. And if these feature sets are not independent, you also build a distribution of the covariance, okay? So you build a statistical model of the variance and the covariance of the model inputs, and then you use the statistical atlas to super sample the distribution and generate a much larger cohort of virtual patients who have feature sets that don't map to any of those measured in real patients, but the range of value is within the distribution, both in terms of variance and covariance, okay? And then for each of these virtual patients, you run your digital twin, but this time, because you have a much larger cohort, you have a prediction, a response of a large population, and you can, for example, do a classical interventional randomized clinical trial where you compare, for example, the placebo, which means do nothing, and see how many patients fracture over 5 years or 10 years, to a certain treatment, and then see how this number of fractures is reduced over a certain time because of the treatment. So to move from the digital twin to an in silico trial, the first thing we need is to generate a virtual cohort following the technique that I just described, which we did. This is work done primarily by Nino Lamattina in my group, and Nino developed a fairly sophisticated statistical model that transformed 100 cases of the Sheffield cohort into a virtual cohort of 1,000 virtual patients, okay? Typical number of a phase 3 clinical trial. I could discuss a lot about the subtlety, but the paper about this just came out. It was accepted a few days ago, so we should have we got the proof yesterday, so it should be out in a few days and you will find all the detail there. The second thing you need to model is how the disease will progress in time, because what the digital twin gives you is the picture at time 0, but then the osteoporosis will continue to progress. And if the patient is not treated, the bone mass will continue to decrease over time. So how do you model this? Well, there are many ways, but the first step we did, and this was mainly done by Sara Oliviero, was a systematic review of 28 large-scale clinical studies that included a placebo arm. So we look at a total of over 27,000 patients that were measured repeatedly over time without any treatment. So to see how the disease would progress, how the bone mass would decrease over time. And we found a fairly complicated distribution, but the good news is that the rate is quite constant. There is a good linear trend, okay? So we can model with a constant rate, which vary by subject. There is a fairly large inter-subject variability. So this should be another stochastic variable that we add, and every member of our virtual population will have a resorption rate, which is greater or lower according to this distribution identified experimentally. Now, with this information added to the model, we can now run the so-called placebo trial. So observational trials where you take 1,000 patients, you just observe for 5 years, and at the end of 5 years, you count how many of this patient had a hip fracture, okay? There are a number of studies like this that are documented in the literature, and what we are doing right now is to compare our virtual cohort with the actual clinical placebo trials to see if there is good agreement, okay? Okay, how much does it cost, the placebo simulation? Well, if you go brute force, you need 100 core hour times 1,000 patient times at least 10 time steps, assuming a 10-year simulation and 1 year time step. So we're talking about 1 million core hour. Now, I will use as a reference the new supercomputer that is coming to Bologna soon, hopefully. No, actually it's already there, but it's not in production yet, Leonardo. It's one of the 2 pre-exascale machines installed by the Euro-HPC consortium. And so say that we have all the 5,000 nodes for this type of simulation available in Leonardo, and assuming we can do 16 jobs per node, it would take about 12 hours using the whole supercomputer, which is a lot, okay? So we need to think a bit of a smarter approach, and again, this is work by Nino Lamattina. We came out with a different simulation approach using Markov Chain Monte Carlo. So essentially, we start with 1,000 patients, and then we simulate every X time falls, and those who fall and fracture are excluded by the simulation. Those who don't fracture keep going, and eventually they might have another fall, and then when they fall, we run the simulation, and if they fracture, we exclude, and so on. Okay. Now this save time. It can be a lot of time if you have a lot of people who fracture early. Not so much if you have a lot of people that don't fracture early, but looking at average condition, we can estimate that we are in the ballpark of less than 200,000 core hours per 10 realization, because of course, being a statistical model, you cannot run once, you have to repeat the simulation a few times to have statistical credibility. So we're talking about probably a 7-hour on a more reasonable 2,000 nodes on Leonardo, which still has a lot of computational power, but something a little bit more reasonable. But we're still missing the treatment. Until now, we can simulate the only patient who had no treatment, but what we want to actually simulate is the treatment, okay? There are essentially 3 ways we can simulate the treatment, and they are progressively more and more complicated. So the first is that we still use the same phenomenological representation we use for the disease progression, but we simply say, okay, because this patient is being treated, instead of losing bone with this rate, it will lose her bone with a lower rate, or it will not lose bone, or even it will gain bone because of the treatment, okay? So we just introduce the treatment not by describing mechanistically how the drug works, but simply in terms of its effect, okay? Now, this worked very well if you want to simulate drugs that are already on the market and who is being extensively tested with large clinical trials, because you can see the difference between the placebo and the treatment and choose the parameters, okay? And this is what we are doing now. The other thing you can do, and this is something we are working on, and I'm not sure it will give reliable result, but in some cases might be the only option. If you've done animal studies, so you know how much the drug reduced the resorption rate, but not on patient, on animals, you can try to scale this to humans, okay? How? By scaling the placebo response that you have on human on the animal model, so say a mouse or a rat. So see how the placebo scale and assume that the drug scale equivalent. It's a huge assumption, which might or might not be true, but that would give us the possibility to do this type of simulations also for new drugs, which are being tested only on animal, but not yet on humans. Otherwise, you model the drug. Of course, to model the drug, mechanistically, you need to have a good understanding of the mechanism of action of the drug, which is more and more the case, but it's not always the case, okay? So in some situation, we will probably force only to option 1. But if you know what is the mechanism of action of the new molecule that you want to simulate, then you have 2 possibilities. The first is that you describe a small compartment, typically in the order of some 100 microns of side. And within this compartment, which is called the bone remodeling unit, you describe the change of the number of cell types and of the diffusion reaction of a series of biomarkers and proteins and the drugs and the inhibiting effect of the drug on certain pathway, not other. And you describe all this as a system of ordinary differential equation. So essentially, you have quantities that change over time using ODE models. There are already this type of models available in the literature. For example, one that I like particularly is being developed by Peter Pivonka, an Australian colleague. And you know, you can imagine to push this to describe something in the order of 1 cubic millimeter, and that would map to a single finite element, okay? So if you have 1 million of finite elements, you can couple to each of them an ODE and predict how the drug will change the resorption in that particular volume of tissue, okay? The other approach is to use agent-based models. Agent-based models are more elegant to some extent because they allow the concept, they bring the concept of space. So you might, for example, decide that a certain cell change status depending on whether it's close or to another cell, which is relevant for bone remodeling. So, but it requires, of course, much more sophisticated model. There are a few implementations around, but none of them is being extensively tested yet. Now, the phenomenological model, the first one, is obviously the simplest to implement. Essentially, we already have implemented. It's just you need just to change a number and rerun your simulation. And it makes very easy to do this for existing drugs. So this is the second level of validation. After demonstrated that our placebo arm reproduced clinical trial on placebo arms, the next will be to take drugs that are being extensively tested in a clinical trial and show that our model actually reproduced, our in silico trial reproduced those results. The animal scaling things, it's a big question mark. It might be unreliable. We need to test. Still, it might be the only option if you have no clue what is the mechanism of action of the molecule. The ODE, you need to run a lot of ODE. Like, you don't need to run 1 million, probably. You can use some surrogate modeling techniques, but you know, you need to run a lot of ODE. But this is the classic parallelization problem. You have the same system of equation, you only change the initial conditions, okay? And so there are very efficient implementation on GPUs that can solve on a single GPU node even 5,000 ODE systems or even more in some cases. And so few back envelope calculations suggest that using the strategy would probably add something 20 core minutes to each time step to our placebo simulation. And what about agent-based models? Well, the computational cost of agent-based model depends on the number of agents, and this in turn depends on the cellularity, so the number of cells that are present in a unit of volume. Bone marrow and bone tissue have very different cellularity. I mean, bone marrow is full of cells. Bone tissue, mineralized tissue, has a very small number of cells. So this is a good benefit because essentially only a portion of the compartment has a significant number of agents. Back envelope calculation suggests that the typical finite element with an 8 cubic millimeter volume contains around 4 million cells. And for a typical ABM solver, that would mean about 80 minutes. But there are some very clever multi-GPU solver for agent-based models that are appearing. And then I think we can push down the envelope around 20 minutes per core, per time step. So in other words, ODE or agent-based, in the end of the day, the computational overhead is not that different, okay? So we are exploring both implementation, but for our discussion, doesn't really matter which one, okay? So here we are, we have finally a full in silico trial. So we have a finite element model that predicts the strength. We have a full model that predicts the input force. We have our virtual cohort that we develop in collaboration with Flinders University. We have our bone remodeling simulation that simulate the effect of the drug. We can run a full phase 3, 1,000 patient, first treated with nothing, so the placebo, and then the same patient treated with the drug being tested, and see how much the drug reduced the number of fracture observed in 5 or 10 years. And you know, here comes the advantage of in silico trials. For example, you can treat the same patient twice with 2 different drugs or with placebo and the drug, something you cannot do in reality. Okay, you cannot treat twice the same patient in a study like this. The other thing is you can do a 10 years simulation at not much bigger cost than a 5-year simulation, whereas double the duration of a clinical trial has a huge cost. So here are the benefits, of course. So putting all together, what do we need? Well, we need about 17 hours on 2,000 nodes of the new Leonardo pre-exascale machine back envelope. This is a lot of computational time, okay? It's not something you can have a normal cluster. You need supercomputing system to do this. You need HPC. What can we do with this technology? Well, today the technology is not qualified with regulatory authorities. So it can only be used in activities that are not of regulatory relevance. So early activity in drug development, or as tool to support the design of the clinical trial, to choose the parameters for a clinical trial, okay? But we are engaged in a very extensive conversation right now. We had our main meeting yesterday with the European Medicine Agency and with the Food and Drug Administration to understand what we need to do in order to qualify this methodology. It sounds like we need to do a lot of things, so it might not happen tomorrow, but when it will, then this technology could be used practically over the entire life cycle of the drug development. What do we need to do to get something like this accepted from a regulatory point of view? Well, for medicinal products, there are currently no established standards. So everything I'm going to tell you, it's my opinion, informed by some informal conversation with officer at FDA and EMA, and some preliminary interaction we had with the authorities. So take what I'm going to tell you with some grain of salt, okay? So the idea of the regulator is, well, I don't care if this is a model. I treat it like any other technology that produces a biomarker. So I'm not really interested into the technology itself, its precision, accuracy, but I just want to understand if that biomarker can be used, is reliable to support the regulatory decision, okay? This requires that if you want to use this technology in support of evidences of new drugs, first you have to qualify your methodology, okay? And this process is called qualification of new methodologies. It exists both in Europe, the European Medicine Agency, at the FDA in the CDER, the unit that follow, the division that follow medicinal products. Conceptually is quite similar. You go and you produce evidences that show that that methodology is good enough to inform the regulatory decision. If you convince the regulator make public a qualification opinion, and from then on, anyone, not only you, but anyone can use that methodology in their clinical trial without any question on the methodology itself. So we started to explore what is necessary to get qualification, and we didn't discuss about this big picture that I represented you. We made a very, very, very humble context of use, okay? So can we use the digital twin, the RF0 predicted by the digital twin as a response variable in multi-dose phase 2 studies? Essentially, these are early studies in phase 2 where you take some patient and each patient you give a slightly different dose, and then you see which dose produced the best response. And you cannot use fracture because these are relatively short studies. So typically you use RLBMD here, bone mineral density. So our suggestion is, given that RF0 is more accurate than BMD, it should also be a better response variable. And what we're being suggested is that we should separate completely the technical validation. And for this, I have to say they're quite open to the idea that we use the new technical standard recently proposed by ASME, the ASME VMV40, even if this starter is not targeting drugs, it's targeting medical devices, but they're not concerned with that. They said, okay, you want to use the VMV40, you can use it to demonstrate that your predictive model is technically valid, and this is essentially is a VV-UQ evaluation, okay? And the only difference is that before the VV-UQ, you do a risk analysis, and you need to evaluate what is the impact in case your model is wrong, and how much the model influences the final regulatory decision, okay? And you have to quantify these two information, and then you have a level of risk, and depending on the level of risk being higher or lower, you choose your VV and UQ plan. So if the risk is low, you can make things a little bit simpler. If the risk is high, you have to go a very thorough VV and UQ plan, okay? But then these are things I'm sure most of you are familiar. You go through the classical moves. So you have to do code verification. In our case, we're using a commercial code, ANSYS, for the finite element analysis simulation. And ANSYS comes already with a lot of benchmarks, and so code verification comes for free in a way. Then you have to do calculation of verification, which typically are convergence study, mesh refinement convergence. We use a surrogate model, so sampling frequency convergence study, and so on. And then there is the validation, and this is everything I showed you before, the cadaver experiment, the retrospective clinical studies. All of this comes into the equation, you know, and you get your validation. But that's not enough. And actually, honestly, they don't care about that. You have to produce because they have to presume that the technology is technically valid. But what they really care is what they call the clinical validation. And the clinical validation doesn't really consider the fact that you are predicting something. It's just a biomarker. It might be a completely phenomenological quantity that is just correlated with the phenomenon of interest. And so the way they validate actually assume implicitly this, okay? So this can be very complex and also very difficult to apply to predictive models. And usually the evidence they want is that you run a trial where you predict what you observe and then you compare. So in the case that I suggest, that we would have to run again at those escalating those clinical trials for the drug that is already on the market, and then compare the results, the prediction of the model. And that is not the type of clinical trial that is easy to fund, because pharmaceutical companies don't have much interest to retest a product that is already on the market. For example, what if a different result comes out? So this is complicated, okay? So I'm a little bit short of time. I need to accelerate, but this is still an open page of discussion. What about money? Well, here comes the huge opportunity. I'll give you only a ballpark. Back envelope calculations suggest that a full in silico trial with BoneStrength would cost around 200,000 euro between software, hardware, and everything. An equivalent clinical trial on average costs $21 million. So the day we could at least partially replace human experimentation with computer modeling simulation, the economic factor is not a problem. It's definitely convenient. Okay, close to the end. So when this stuff is going to be available for use? So we're finishing the digital twin. We're just doing the last touch. It should be available for use in the last quarter of this year. And we still have a few things to do on BoneStrength. The In Silico trial, we expect to have the finished thing in the third quarter of 2023. In conclusion, I think digital twin can be transformed into In Silico trials, but at a considerable computational cost. And so the scalability of this solution, the possibility to have this solution running on high performance computing system is indispensable, especially if your target is eventually to simulate phase 3 clinical trials that involve thousands and thousands of virtual patients. Before I stop, I would like to thank all the people that contributed to this work in my group: Cristina, Sara, Alessandra, Nino, Chiara, and 2 colleagues from Sheffield that did a lot of preparatory work when I was there, Binaki and Shannon. Thank you very much.
Fantastic. Thank you very much, Marco. Just a quick reminder for the audience, if you do have any questions, then please navigate to the Ask the Staff a Question panel, which should be accessible via the question mark icon on your screen that looks like the one that's being displayed currently. And go ahead and type your questions in there so that I can read them out for you to Marco. So just whilst the audience gathers their thoughts, I had a few questions of my own for you, Marco. Well, I mean, first of all, it's more of a remark, but so these talks with the EMA, and they happened just yesterday, right? It's very interesting to me to hear that they're already open to deploying these types of in silico trials, even if in a sort of informal capacity right now. It's interesting to hear that there's an appetite for that.
Yeah, we need to be careful. So what we had yesterday was a meeting that concluded a process formerly known as qualification advice. So essentially, we went to EMA and we said, we would like to use this in silico technology, and this is the way we would validate it to convince you that it's good enough. And then they told us, oh, we disagree, we think we should do different things, okay. Yesterday there was this meeting where there was a very intense discussion between us and the expert of EMA, and soon they will send us a written reply, which is the qualification advice. And we are expecting that what we proposed originally was rejected, it's not good enough, and what they want is a full-scale clinical trial, interventional clinical trial, as I mentioned earlier. So the good news is that EMA is willing to discuss seriously about this. The bad news is that probably we need to choose the right application, because for some application, the level of evidence and the experimental activity required to obtain it might be too expensive. And so it won't be possible to do this in a short term. But yes, good news. I mean, it's getting real.
Sure, I understand. No, that's great news, absolutely. So you made the point that computer simulation techniques are widespread across just about every other industrial sector. What do you think is the main reason for the slow adoption in healthcare?
I think there are 2 reasons, one pragmatical and one more cultural. The pragmatical is that living organisms are incredibly complicated. And until recently, we didn't have the math. And even when we had the math, we didn't have the computational power to solve that math. So mechanistic, biophysical, biophysiological models tend to make very crude idealization of real processes. And as a result, they were usually very inaccurate, okay? Now, I think, you know, the big change in computational power and the sophistication of computational science and engineering has reopened the game, in my opinion. But then there is a cultural reason, and the cultural reason, allow me to make a little historical digression. I mean, science as such started in the 17th century, and at a time where the general culture was very strong around a dualistic ethos. So there was the matter and the spirit, okay? And scientists had to be very careful not to impinge into the spirit side, okay? So physicists, what we would call now physicists, focus on inanimate matter. And they were free because inanimate matter doesn't have soul. So in a way, they were freer to you know, express things in a truly mechanistic way without worrying too much about that. The scientists that we call biologists today had to be a lot more careful. So the whole biological science developed for many years around a very phenomenological agenda where biologists were observing and describing, but not really explaining in a quantitative, mechanistic way. So this has created this parallel life between physical science and biological sciences. And this gap remain, even if, of course, meanwhile, this gap is being filled in many, many instances. But culturally, I think there is still this separation that have very old historical roots.
I see, I see. And do you think, when we achieve exascale, or at least widespread exascale, do you think that might close that gap a little bit?
I don't think we can oversimplify. What I try to explain today, and that's why I didn't speak only about HPC, but I speak also about things that seems not to related, like regulatory barriers, etc. The acceptance of this technology will not only pass through our computational ability to solve huge problems, but that ability is definitely very important. So it will help a lot. I'm sure about it. Because we will be able to make much less idealizations, and that will make our model much more credible and easy to explain and easy to validate. So yes, it will make a huge difference.
Of course, of course. I've got a question from the audience here, actually. This is coming from Martha de Cunha Malef Bergman, who not only says fantastic presentation, very insightful, but also asks a question. So towards the beginning of your talk, when you described the Bologna and Sheffield cohorts, they consisted of only postmenopausal women. But Martha is asking, what about the male population? Does your study also consider male cohorts?
Yeah, good question, thank you. So historically, osteoporosis was a female disease because women start to have osteoporosis even just after menopause. So you can have severely osteoporotic women even in their late 50s or early 60s. Okay, and because the life expectancy was not that long as it is today, that was it. Okay, now that we have significant number of male that survive until their 80s, even 90s sometime, we do have significant number of osteoporotic fracture also in males. And but this population is definitely underrepresented in all studies. The results we have would suggest that there are no fundamental differences in the biophysical process of falling and fracturing. Of course, there are a lot of differences in the underlying biology of bone remodeling, but the biophysical aspect that I described today should be quite similar between male and female.
Sure, I'm with you. And then I had one more question actually, is how reliable is that inverted pendulum model? Because for example, patients, they might use their hands to soften the landing, for example. How true to reality is that assumption?
That's a very good question. The point is that there is no point in making a very detailed, sophisticated model, because then you would not have the experimental observation to inform that model. I mean, for other application, we have whole body, multi-body dynamics model. So we could simulate every detail of the fall. The problem is we don't know how the patient will fall.
Sure, okay.
So what we did is to use an extremely simple model, probably oversimplified, I agree with you, or better, the inverted pendulum give you the upper boundary of the impact force. And then to bring all the other factor as stochastical variable, in particular, this postural attenuation coefficient that we define phenomenologically and using a statistical distribution, account for all the compensatory strategies like slowing the fall by putting a hand first or bending your knees, which means you lower the center of mass while you're falling, and falling against the wall before hitting the floor, and so on and so forth. Now, we could model all this with great detail. If you ask me to model one of those falls that I show you, I can do it with great detail. But in the future, I don't know how my patient will fall. So we need to accept that, you know, the crudeness of the model has to be compensated by this stochastic simulation. That's why in the digital twin we simulate million folds to look for the worst case scenario.
Right, I see. So this inverted pendulum in some senses represents kind of the worst way in which a patient could fall, given no intervention by that patient.
Yeah.
Right, I'm with you. Okay. Well, that was all of the questions that I had, and I'm not seeing anything else coming in from the audience. So I think this might be a good place to draw the session to a close. So if you wouldn't mind going over to the next slide for me, Marco, and I'll give the free scalability service a quick plug. So before we end, I'm just going to talk about CompBioMed's free scalability service. So if you are someone who uses or even develops biomedical applications that necessitate intensive computation, but you're running into performance issues that are limiting the scope of your research, then CompBioMed offers a service completely free of charge to improve the turnaround of your computing jobs. Next slide, please. So if this is something that applies to you and your research, then you can fill out an application form available on the webpage displayed at the top of this screen. Otherwise, you can also feel free to reach out either at the email address shown here or via the In Silico World Slack channel, which brings me on to the next slide. In Silico World is a community devoted exclusively to in silico medicine, offering expertise, opportunities for collaboration, and also a safe space to exchange ideas and advice. Next slide, please, Marco. Already, In Silico World has a large following, which includes members from large biomedical companies, biomedical SMEs, independent software vendors, clinical research institutions, and even regulatory and standardization bodies such as the FDA and NICE. So that's all I wanted to say on that. Thank you once again, Marco, for giving us your time today. A reminder that this e-seminar will be available on the CompBioMed YouTube channel and website, which is accessible via the link shown on the next slide. Yes, that's the one there. Perfect. Thank you for that, Marco. So finally, thank you to everyone in the audience for attending, and I hope that you enjoy the rest of your day.